Hi, I'm OpenMx, and I'm delighted to introduce the topic of modeling and estimating genetic and environmental components as part of the 40th International Statistical Genetics online workshop. This topic will be introduced in 4 parts, starting with ways to estimate heritability, followed by how to do this using data from relatives and in particular twins. We will then model genetic and environmental sources of variation in a single trait and in multiple traits. As my presentations build on those of my colleagues, I'd like to give them proper credit. So let's start with part 1, ways to estimate heritability. Here is a brief outline. We will first recap some quantitative genetics theory, refresh the concept of genetic relatedness, reintroduce the Haseman-Elston regression and its relationship to the classical twin design. We will then shift gears and introduce a simple linear regression model, as well as the method of path analysis, to show how we can go from an equation to a path model, and then eventually to an OpenMx script that we will use to estimate heritability from twin data. We will provide a brief introduction to OpenMx and show how we fit the same linear regression model in OpenMx. So let's start by recapping some quantitative genetics theory. As you'll see, my slides all start with a question mark that helps guide its contents. So what is heritability? It's the proportion of trait variance explained by genetic factors. Thus, if a trait or phenotype Y can be modeled as Y equals genes plus environment, then heritability, often denoted as H squared, is the genetic variance divided by the total variance. Alternatively, heritability can be derived from the phenotypic correlation between relatives and their genetic relatedness, RJK, using the following theorem, where RJK is the coefficient of genetic relationship between individuals J and K. So heritability quantifies the degree to which inter-individual differences and resemblance in populations are due to genetic factors. And it can be approached in 2 different ways, either by looking at differences between people in the population or by the resemblance between relatives in families. This fundamental theorem is used to estimate heritability using individual level SNP data in Haseman-Elston regression or Gremmel methods. So how do we estimate genetic relatedness from SNP data? The coefficient of genetic relationship is defined as twice the probability that 2 alleles picked at random in individuals J and K are identical by descent. And it's these coefficients that make up the genetic relationship matrix and can be quantified based on either expected IBD sharing or using actual SNP data. One of the standard estimators of this coefficient of genetic relationship is implemented in the software GCTA and referred to as pi hat. The expectation of this pi hat is that it is exactly the coefficient of genetic relatedness, as shown in this figure, which also shows that the observed relatedness actually varies by relationship. So how do we use this genetic relationship coefficient to estimate SNP heritability? We can use the Haseman-Elston regression, which is a classic statistical genetics tool that relies on the association between phenotypic similarity and genetic sharing. The original model regressed the squared difference in siblings' traits on the proportion of markers shared IBD to detect linkage. It was extended to use cross products, which increases the statistical power. And then it estimates the heritability by regressing the phenotypic similarity, is expressed in this correlation between normalized residuals, onto the genetic similarity, the pi hat that we've just defined. So here is the formula of the cross-product method, which looks exactly like a simple linear regression, where the beta-1 coefficient directly estimates the heritability, which is shown in this figure here. And this method is rather similar to the assumption-free estimation of heritability from genome-wide IBD sharing between full siblings or twins, as developed in Sarah Medlin's group at QIMR. Now, how does SNP heritability relate to twin heritability, which is the topic of today's presentations? They both have the shared goal of partitioning phenotypic variants to estimate heritability by analyzing how phenotypic similarity between individuals relates to their genetic relatedness. With HE regression using DNA-based relatedness, whilst the twin design uses inferred pedigree relationships. So how do they differ in their estimation of heritability? As we have just seen, the Haseman-Elston regression regresses the phenotypic cross products of siblings or of pairs of unrelated individuals on their shared proportion of alleles estimated either via IBS or IBD, using this formula. The classical twin design, on the other hand, which is the traditional method, compares monozygotic or MZ twins to dizygotic or DZ twins to partition the variation in a phenotype, not only in sources due to additive genetic factors, but also to common environmental and unique environmental components, which can be expressed into this following formula, where we can get an estimate, again, by relying on the relatedness, in this case, of MZ and DZ twins, and also estimate simultaneously the variance due to shared or unique environmental components. Now, why may the estimates of SNP heritability and twin heritability differ from one another? Well, these methods make quite different assumptions. The classical twin design relies on the equal environments assumption, more about that later, as well as the assumptions of zygosity, or genetic relatedness, whilst the HE regression uses DNA markers to calculate the GRMs to estimate heritability, and it thus requires that the genetic similarity measure, the coefficient of relatedness, truly captures the genetic variance without confounding by shared environmental factors. These methods also differ in their effect magnitude, where the classical twin design often yields higher heritability estimates than HE regression based on SNPs. One of the reasons may be that they're measuring different components of the genetic variance. In the rest of today's presentations, we will provide a lot more detail about the classical twin design. But before we do that, we want to introduce some of the tools that we're going to be using throughout. And we do that by illustrating how you fit a linear regression model with one SNP in R, and how we go from this simple linear regression in R to a structural equation modeling approach that we will be using primarily when we conduct a twin study. I assume you're all pretty familiar with a simple linear regression where we predict, for example, height from a SNP or y from x using this simple equation, where y is the dependent variable in participant I, x the predictor in participant I, and e the residual. And we're interested in the parameters b0, the intercept, as well as in b1, the slope or the regression coefficient. Trying to address the question, are x and y linearly related, where the null hypothesis is that the b1 parameter is equal to 0. Now, how can we do this in R? We can use the simple LM procedure. But let's first look at the descriptive statistics here of both height and the SNP. We can show this graphically in a histogram and show the regression using by plotting the number of alleles So let's start by showing how to do linear regression in R, where our question is, does the SNP predict height? Here are the descriptive statistics for height and the SNP, which is coded additively as 0, 1, and 2, indicating the number of big A alleles. And here's the one-liner in R using the LM function to regress height on the SNP. This is the typical output of LM, and it allows us to answer the question, is there a significant association between height and the SNP, or does the SNP explain variance of height? In this case, the answer is yes, because the p-value is less than the critical value, and this is a linear additive model such that the effect of the allele, the big A allele, is additive, as shown below. Before we turn this into a structural equation or a SEM model, let's introduce the method of path analysis. As it provides a rather intuitive way to present a model. So how do we specify models using path diagrams? Here is just an example of a path diagram where we follow a certain number of conventions. So typically squares or rectangles, such as P here, are used to denote the observed variables, while circles or ellipses denote the latent or unmeasured variables. We use single-headed arrows or paths to represent causal relationships between variables, where the variable at the tail has an influence on the variable at the head. We can also use double-headed arrows to represent the covariance between two variables through common causes that are not modeled. It is also used to represent the variance of a variable. You may not be as familiar with either the triangle, which denotes means, or the diamond, which denotes definition variables. More on those later. To be able to read a diagram and to derive the expectations between the covariance between different variables from such a diagram, it's important to follow some very simple but strict rules. And these are that you can trace backward, which means going from the head of an arrow to the tail of an arrow, change direction in a double-headed arrow, and then trace forward. But it's important that you can't trace through two-headed arrows in the same chain. In other words, you can't go back and forth through a two-headed arrow. How do we then get the expected covariance between two variables? Or get the expected variance of a variable? We do that by multiplying together all the coefficients that we can walk in a chain and summing over all possible chains. We will provide an example of this in the practical during the workshop. So now we're ready to go from the equation to a path model. And here we do this for our simple example of linear regression. So you're all familiar with the equation. We're going to turn this into a path diagram that will then allow us to work out what the implied means are and the implied covariance structure from our linear regression model. This can be done in two ways. The first one is the one that is most directly related to the equation, but requires the use of this definition variable, which is a variable that we can put on a path so that we can multiply our parameter with the value of that definition variable. And so this particular diagram then translates into the expected mean, which is calculated from the triangle to the dependent variable height here as b0 plus b1 times the value of the SNP, and maps on directly to the equation from the linear regression. In addition to the effect of the SNP, there is also some residual variance, so that is included in the expected variance of height. Alternatively, we could have specified both the SNP and height as two observed variables, which would then imply that we have to specify the expected means for both height and the SNP. In this case, of course, for the SNP, that is simply x. And when we then work out the expected variance, in this case, we have two variables, so we have the expected variance covariance matrix with on the diagonal the expected variance of height as well as the SNP, and then in the off-diagonal element, the expected covariance, which in this case is b1 times the variance of the SNP. Now that we know how to read a path diagram, let's provide a quick introduction to OpenMx. This is taken from the OpenMx website. What I do here is basically just introduce very simply the kind of structure or outline of a script that we will be using throughout, where we start typically with a little description of what our script is doing. We then prepare the data. We have then a section that prepares the model, and then finally a section where we build the model and fit it to the data and provide the summary statistics. I've also included here some of the core syntax that we will be using. These are all the MX functions that are built in once you load the OpenMx package. So let's look at our first OpenMx code. How do we specify this linear regression model in OpenMx? So this is our model. As I said, we first have the data. which we read into an MX data object. And then basically OpenMx is a matrix interpreter. So all the data as well as the model are going to be specified using matrices. Remember that we wanted to use the SNP as a definition variable, which means we need to put it into a matrix using this special formulation data.snip. Next, we have the model section where we create matrices for each of our parameters of interest, the B0, the B1, and the E parameter, and each are put in matrices that are defined by their type, the number of rows, columns, whether or not it's a freely estimated parameter, what the starting value is, and the name that we want to use, and that we then also use when we want to create the algebra that defines the equation, which is done here in the expected mean and the expected covariance statements. And you'll see that the algebra in OpenMx maps on directly to the equation above. We then have some other functions that allow us to combine the model and the data, indicate which fit function we want to use, and then put all these objects together in the model that we can then run and generate the output for. Now, this first model includes both the B0 and the B1 as free parameters, but to test the significance of this B1 parameter, we need to drop it from the model, which we will do here after calculating the R squared, again using another function mxeval, and then we use the OMX set parameters function that allows us to fit a simple submodel where we fix the B1 path to 0 and then look at whether or not it significantly deteriorates the fit of the model. Now you may wonder, why do we need 15 lines when we can simply do this in a one-liner? That's a very good question. Before we go there, let's show you that the results from our single lm command in R actually match those of OpenMx. So this is your typical output from the R code, where I've highlighted some of the numbers that are of most of interest. And then we have the output from OpenMx, where you see that the estimates of the parameters are almost spot on, as are the estimates of the R squared and the significance level of dropping the beta 1 parameter from the model. So you may ask, why not OpenMx? As it seems to be much more complicated than what we can do in R. We know it looks complicated, it has a steep learning curve, but it also has a number of advantages that I'm not going to read for you, except for the fact that it is powerful and flexible. It is free and open source. Scripts can be put into functions just like LM, so they become a one-liner. And the software was developed with genetically informative data analysis in mind, and so there's a lot of code out there to do the typical kind of models that you would fit when you have access to data from relatives. Thank you.